what happens when one of these machinery gets it breaks down, for example?
Yeah. So this is what we look at. So there is, I have to say, the field of development and the field of ubiquitination, they are not yet, they are not yet talking to each other. Now and that's why we're very excited about this, because we're, very interested to look at the developmental context of a lot of modus operandi of these molecules, right? And so ubiquitin gets conjugated to a protein in a very complicated process. Well, not very complicated, but it's a cascade reaction that involves enzymes of different classes, and ATP, as you mentioned, and magnesium, and then there's but essentially the last step of that reaction is catalyzed by enzymes, so by proteins that have a kind of a, I don't even know how you would define an enzyme in that friendly way, right? It's a protein that enables a reaction, right? So
speeds up the reaction.
Catalyzes the reaction. So you'd have proteins that are called E3 ubiquitin ligases. So again, like an unfriendly name, but we call them E3s just for simplicity. And these E3s recognize the protein that needs to be tagged by ubiquitin, and ubiquitin itself brings them together and links the two. And so it happens that we, in the human genome, have over 600 of these enzymes, of these E3s, and they have obviously quite a spectrum of specificities. So each ligase would not only see one protein, but many. Some see a lot, some don't, right? And it is fascinating that if you look at the genetic connection between the E3 genes, so the connection between E3 genes and human disease, we see around 15% of all these E3 genes are directly linked to the neurological disease, which to me is really mind-blowing. But it also demonstrates that for some reason, development or neurons, or both, neural development, really depends on that system quite heavily, right, for it to work well. Otherwise, there would not be the 15. I mean, 15% is a lot in biology, right? And basically, what happens in a lot of these scenarios, and they can be different scenarios that, mostly genetic mutations in E3 genes. I'm saying genetics like 40 times, but the substitution. So they lead to loss of function of E3s. So then the protein that was supposed to be degraded now is not, because the E3 cannot bring the ubiquitin on it, so it doesn't have a way to catalyze that conjugation, and that in turn. So if that ubiquitination was of canonical type, and it was supposed to end up at the proteasome, that causes an accumulation of targets of that given E3s. And this is kind of what also my lab has been doing for many years now, is a hallmark of some of the neurodevelopmental disorder, the accumulation of substrates that rely on the degradation pathway that is mediated by the E3.
Which diseases, if you may say?
So, my lab particularly studies the developmental scenarios in autism spectrum diseases, for example, or intellectual disabilities. This is a broad heterogeneous group of disorders. Quite now in the realm of modern genetics, we can narrow down the disease type. And one of them, for example, that we have published in 2021 was Kaufman oculocerebrofacial syndrome, so KOS, K-O-S. And this is a rare genetic condition or disease that affects kids, and it affects them quite profoundly. So there's developmental delay, there's autism-like features behaviorally, but there's really dramatic kind of brain image where the connection between the left and right hemisphere is very thin. The ventricle in the brain is dilated. There is obstruction to the cerebrospinal fluid flow through the foramen magnum. So there's a lot of kind of structural issues in the brain of the kid. So there, what we have done is, we have, and the cause for that disease is the mutation in one of the E3s. The gene is called UBE3B, and what we have done there is this was one of my PhD projects, and we have really chased down what UB3B ubiquitinates in the brain and where, and then kind of narrowed down the cascade that might be associated with that disease in humans.